Hello and welcome to this webinar titled Characterization of Subvisible Particles in Protein and Viral Vaccines, hosted by Biopharma Asia magazine and presented by Marina Kirkitadze, Head of Process Support and PAT Platform Analytical Sciences at Sanofi Pasteur, and Bonnie Edwards, Product Manager of Imaging and Mfi at ProteinSimple. My name is Stephen Edwards and I'll be your moderator. Now, please allow me to introduce our first presenter. Marina Kirkitadze is Head of Biophysics and Conformation Unit at Biochemistry Platform Analytical R&D North America, Sanofi Pasteur Limited, Toronto, Canada. She has 17 years of experience in the vaccine industry. Marina received her PhD in Biological Sciences at the Institute of Protein Research, Russian Academy of Sciences, Pushchino, Russia, and her MBA from the University of Phoenix, Arizona, USA. Marina's main focus is a characterization of vaccine components, specifically protein conformation and stability in solution and in adjuvanted forms. I will now be handing over to our first presenter. Welcome, Dr. Kirkitadze.
Hello, everybody. Thank you very much, Stephen, for the opportunity to present, and to Adam Young as well. This is a great opportunity to share our experiences with subvisible particles and continuation of the work with Bright Talk and with the Biopharma Asia. So today, the topic, as Stephen alluded, is subvisible particle in the viral vaccines. And if we go to the next slide, that shows the summary of the study. So the purpose of the study is to characterize the size and composition of the visible and subvisible particles in live replication-deficient viral vaccine candidate, which is used against HSV genital herpes in the early development stages. So stress samples of HSV2 vaccine candidates were analyzed to mimic the virion uncoating and contact discharging conditions that were observed during cell disruption. The mechanism of aggregate formation is discussed, and a two-step thermokinetic aggregation profile is proposed. So this is the first time that we share the detailed analysis of the particles in a viral vaccine product, and more details you can see they are reported in the reference provided. The first of is Lillian Lee. She's my colleague from Bioprocess R&D at Sanofi Pasteur, Toronto. And there is also a disclaimer that the views of the presenter do not necessarily reflect all the views of Sanofi Pasteur or affiliates. So on this slide, basically we see the illustration of existing tools that can be applied for characterization of the particles. In the study presented today, we used DLS, Mfi, and TEM, so quite a few. Some of them are particle counting techniques like Mfi, microflow imaging, and TEM can be used for that purpose too, more in the submicron range, whereas DLS definitely goes into the ensemble-based techniques. So on the next slide, basically, on the next slide, you can see the summary of the study, so-called graphical presentation, if you will. So, inherent particles in HSV viral vaccine candidate. So basically, on there we'll see the container of the vaccine, and if you zoom in, you may see the visible particles. Obviously, by light microscope, you can do that. And if you switch to Mfi, for example, which is image next to it, you will see the example of subvisible particles. So HSV contains both. So for that study, we applied a number of techniques. Basically, on one hand, to do rigorous investigation, and on the other hand, is to establish the array of methods that can be applicable for the viral vaccines in the future. whether we use them or, potentially can share with public, and if they may be found applicable for other viral vaccine existing in other industries. So if we move, just to highlight, so the visible particles were studied by light microscopy, then they were collected on a filter, dissolved, used on SDS page, and confirmed. The identity was confirmed by Western blot. The in-gel digestion of the excised bands for the gel were used in LCMS, and by mass spectrometry we managed to also confirm identity of the protein. So basically you see that one technique kind of intertwines in another. On the other hand, you see DLS application and also RALS, which is right angle light scattering. They both were kind of similar techniques, but at the same time we used them to confirm observations made by the first one. With the same respect, TEM and MFI, they're complementary. TEM, you can see the transmission vector microscopy at the submicron range, whereas with MFI on the micron range. So basically subvisible particles all the way to visible range. Okay, so with the next slide, we're going into the presentation of visible particles of the HSV-2 viral vaccine. So on the left-hand side, on panel A, you see the presentation of the obtained by light microscope. So basically, then the particles, as mentioned before, they were also examined by FTIR microscope. Why is that? Is because you can see on the panel B quite similar distribution, but also you see more dense particles just because of the magnification is better. And also you can zoom at individual particles and collect the spectrum. And basically that was used to determine spectroscopically what type of particles we're looking at. So obviously, as you can see from the spectrum below, it contains the amide-1 and amide-2 regions, which is quite typical for the presence of proteins. So it gave us a hint that, yes, indeed, we have potentially proteinaceous visible particles, and so subsequently they have to be studied with other techniques that can confirm identity more explicitly. And so with that, we turn to SDS page Western blot at first. And so with basically on the panel A, you see the marker in the left panel, and then on the right panel, the run. And then basically we collected different fractions. But the point is that each band after that was excised and used of this exact identical gel, excised and used by mass spectrometry to confirm identity of the proteins. However, one of the copy of this gel was used to proceed further to Western blotting, and with that we could confirm that many proteins, they come from the virus itself, from HSV variant, and it was blotted with antiviral host cell proteins and also with HSV-2 monoclonal D antibodies. So basically the conclusion was that visible particles contain the proteins from the HSV variant itself and also from the host cell where the virus was grown. So the mass spec results are not presented here, but if you're interested, again, the reference is provided. The paper is published in Current Pharmaceutical Biotechnology, and the supplement contains the LC-MS data. So basically, two methods confirmed their identity. With that, we move to basically attempt to analyze what is the stability of the variant. And one of the tests that we could do is to use DLS, so dynamic light scattering, to follow temperature-induced aggregation of HSV-2. So as you can, the samples were subjected on one hand for 45-degree exposure for 1 hour, 60-degree incubation for 1 hour, but also to the freeze-thaw cycle. And on the right-hand side, you can see in the panel the DLS results for the freeze-thaw cycle. In both cases, with dark burgundy profile is shown the control samples, and with orange and pale green, the samples that were on the left-hand side, 45 and 60 degree, on the right-hand side is freeze for cycle 1 and freeze for cycle 2. Obviously, there is some change, significant change observed for the 60-degree sample. Some changes are occurring after freeze-thaw. However, if you look at the numbers, the change is really small. So basically, to evaluate that, we also did the, here basically on this slide you see examples of the individual subvisible particles of HSV2 that were captured by MFI, and the particles you see are in the range of 50 to 100 microns. MFI was performed at the same samples as you've seen by DLS, but basically it was done in the range of 1 to 100. However, the particles were counted from 1 to 70. That's just the basically the capacity of the instrument. And so all of them exhibited ribbon-like structures, filamentous, and they varied in size. So with that, we also did TEM, which is on the next slide. So this is both freeze-thaw cycle and thermal stress. And so control samples in panel A, freeze-thaw cycles after the second cycle is shown on the panel B. So you see some changes, which basically consistent with what we observed at the DLS, but changes are very small. And then at the same time, 45, you see changes occur, but at the 60, the majority of changes happening at exposure at 60 degree. While we see the virion rupture, we see basically aggregation forming quite severely. So really, the most significant impact is heat treatment at 1 hour at 60 degrees. And so also main conclusion that we can link here, that the DLS data and TEM data basically allow us to conclude that particle changes in submicron range are related to HSV variant structure itself. So here is the summary slide of something that you already seen, such as DLS and TEM, but also there is MFI data captured. So basically the total concentration from 1 to 70, and then in ranges, for example, 1 to 2, 2 to 10, 10 to 25, and so forth, up to 70 microns. And as you can see, there is no significant difference between the particle count in between control and freeze-thaw cycle. There is some changes, but they cannot be considered significant changes. And so the most important readout, what you can see here, is the lowest row. It's with infectious titer, which is our biological test for the virus. And infectious titer presenting here in a log values basically show that for control sample we see 7.2 versus for freeze-thaw cycle 1 and 2 we see subsequently 7.24 and 7.13. So we consider that's a very good result, because that confirms that although we see some changes, they're very small, and we can say that there is no impact of the freeze-thaw cycle on the viral HSV2 variant. Why this conclusion is important is because if you consider different storage conditions and you want to have conditions that basically retain the size, the structure, and most importantly, biological activity, freezing is apparently a good option for this type of a product. Now, the next slide here is the effect of thermal stress on the particle size, concentration, and infectious titer. And so basically you see again the overlay of DLS results, and at 60 you can see that basically the hydrodynamic radius observed are quite large compared to the control and 45 degree. and that was visible from the graph too. You can also see that total concentration of the particles increased, but not really significantly. We can see for a 60-degree, the biggest change observed by Mfi. Between 45 and control, we can say that it's not a significant change. Now, in terms of TEM, same thing. We see that control sample and 45-degree sample are comparable. However, we have to say that there are aggregates, but aggregates observed in both cases. Whereas at 60-degree, the aggregates are quite large, and we see that it's virion assembly. So, with respect to infectious titer, which is our biological activity test, we see a difference even between 45 and control, and for 60 we don't detect any activity at all. So that's really the measure that there is a limit to his stress, but also it tells us the way the virus can behave in the cell if say the rupture occurs and the DNA is released. So what kind of mechanism that we can imitate in vitro to explain how it works in vivo. So there are two purposes here, stability and the modeling part. So main conclusion here that the changes, again, particle changes in the submicron range, they are related to HSV variant, virion structure, and can be and had direct impact on the biological activity. That's the most significant conclusion. Now, as alluded before, what is the impact on temperatures? What would be other techniques to study? So you've seen just now the application of DLS and Mfi. So on another side, we went further. What we did is continuous temperature ramp. On the A panel, you see the overlay of fluorescence intensity at 340 nanometers and the light scattering at 82 nanometers. And so basically that was measured as function of temperature, and the fact that the overlay basically tells you that fluorescence increases and basically the significant changes occur. On the other side, there is a DLS, just DLS continues for the HSV sample, just to see particle size as a function of temperature. And then HSV2 particles, particle size at different temperatures. So we compared continuous temperature ramp with a discrete at the key temperatures we think where transitions occur. And so basically the analysis or combination of these results made us think that we can apply a two-step thermokinetic model to describe the changes occurring under impact of temperature. And so on the next slide, there is a model discussed. So temperature-induced aggregation was observed by RAL, so right-angle light scattering and intrinsic fluorescence in DLS. And the increase in subvisible particles with the temperature could be fitted with thermokinetic model. And so the step one basically is a curve that covers the range between 45 and 58 degrees, whereas second step is between 59 and 69 degrees. And basically, if one wants to explain what events could occur at those temperatures, so obviously from 45 to 58, we see that the aggregation happening, and also a viral envelope disruption and virion opening. And we've seen it up to all the way up to 60 degrees was very clear by TEM, so we can conclude that. However, at high temperatures, 59 to 69 and higher, the events such as DNA exiting the virion and DNA melting and viral protein and residual protein and DNA aggregation can occur. And that information is known from literature. It's done on the HSV virus by DSC. I believe that's oncolytic HSV virus that was used. And also in-house we did nano DSF, and we had a webinar on nano DSF in September, but on the protein products. But nevertheless, this technique is well described in the webinar. I believe it's 19th of September. And so with that, we found that first peak we see around 45 to 50 degrees, whereas the second peak occurs after 70 degrees. So that also kind of gave us confirmation that we see two peaks as well, two transitions. So that confirms the model. And also the fact that temperature coincides with what's normally occurred for protein melting in the viruses and phages, and with a second peak with DNA. So with that, move to conclusions. And basically, to summarize, the particles changes in the submicron range were related to HSV variant structure and had direct impact on biological activity. So also observed that subvisible and visible particles could induce aggregation of viral product. The infectious titer did not correlate with any trend in subvisible particles. I didn't mention here, but we also did a stability study for 1 month at elevated temperatures, 25 degrees versus 4 degrees, and we didn't see significant increase in subvisible particles at that range. The mechanism of protein aggregation was discussed as a two-step thermokinetic aggregation profile was proposed that you've seen just a slide before. And the approaches reported in this study can be applied for other viral vaccines that exist in the pipeline with in our company or any other company that could be interested to use some of the applications. And if we move to the next slide, I would like to acknowledge and to thank everybody who is on this list. Most of these are my colleagues and my co-authors, but however, I would like to mention Jian, Hamin, Yelin, Arna, and Fernando, who were helping with the study, especially with discussions, providing material without which that won't be happening, and also, interpreting some of the results that we didn't use and we didn't share in the study, but definitely helped us to support the conclusions. And so with that, I would like to thank everyone for listening. And basically, the floor is back to Stephen, who will be introducing my co-presenter, Bonnie. Thank you very much.
Thank you, Dr. Kirkitadze. Once again, I would like to remind our viewers that there will be a live Q&A session at the end of the webinar. Please feel free to send in your questions at any time during the webinar via the questions tab located directly below your webinar screen, and we'll go through these at the end of the presentation. Now, please allow me to introduce our second speaker. Bonnie Edwards is a product manager for imaging and MFI at ProteinSimple, a biotechnology brand. She has a BA in molecular and cell biology from the UC Berkeley. Her background includes small molecule drug discovery, high throughput screening, flow cytometry, and lab automation. I will now be handing over to our second presenter. Welcome, Dr. Edwards.
Thank you, Stephen, and thank you, Marina, for your presentation. As Stephen mentioned, my name is Bonnie Edwards, and I'm the product manager for microflow imaging at ProteinSimple. So I'm going to be talking about characterizing subvisible particle populations with microflow imaging. Just a quick introduction to who we are. ProteinSimple is a part of Bio-Techne. As a company, Bio-Techne's focus is on providing innovative solutions for reagents, assays, platforms, custom manufacturing, and testing services. In our division at Bio-Techne, proteins are the core of our business. We have the largest offering of antibodies in the world through our R&D systems and Novus brands, and we've combined that with the analytical power of the tools we produce here in San Jose at ProteinSimple. So let's talk about proteins. As Marina discussed, proteins aggregate, and this is a primary challenge in biotherapeutic formulation. Proteins and formulations tend to form aggregates because proteins are fragile and they can easily unfold or change conformation so that they end up clumping together. As they increase in size, They form insoluble protein aggregates, which can appear as translucent particles, and they can grow from nanometer to micron-sized particles that are described as subvisible particles. Subvisible protein particles, because they are translucent, can be hard to detect and measure by traditional methods of protein characterization. So regulatory agencies such as the FDA have noted that high levels of particles in the subvisible range may be considered a risk to product safety. And while the FDA doesn't have a pre-established limit for subvisible particles, they do consider lower to be better, and they expect that particles should be characterized in order to aid in method development, selection, and risk assessment. To highlight the importance of particle detection, in 2016, particulate matter was cited as being responsible for 27% of the drug recalls that year. So being able to identify particle contamination and protein aggregates early in the process can be critical to avoiding costly recalls down the road. And to address this, ProteinSimple offers a tool for detecting and characterizing subvisible particles. Microflow imaging, or MFI, combines the capabilities of digital microscopy with microfluidics. The MFI 5000 series instrument that you see here on the left provides imaging-based analysis of particles in biotherapeutic products. And our Bot1 system that you see here on the right provides an automated solution for increased throughput. So as I mentioned, MFI works by combining the direct imaging capabilities of digital microscopy with the precise controlled microfluidics. The sample is pulled through a flow cell, which guides it past the camera and optics and keeps the sample within the focal depth of the optical path. As the sample moves through the flow cell, the camera images the entire field of view, and then our software isolates individual particles from that image. The direct imaging capabilities of MFI allow you to gain precise information about the size and count of particles in your sample. So MFI's capabilities are calibrated for the subvisible range of particles that are important for developing therapeutic products. The subvisible range is typically defined as particles ranging from 1 to 100 micron in size. This range includes the particle size bins that are regulated by USP 788, which has set discrete limits for particles that are greater than 10 microns and greater than 25 microns in size. It also covers the 2 to 10 micron range for which quantitative data is needed, but no threshold has yet been defined. And by imaging particles directly, MFI can quantify morphological information about these particles. MFI measures 10 different morphological parameters for every particle that is imaged. These parameters include size, shape, and translucency, and these parameters are used to distinguish between different populations of particles in a sample. So this chart here provides an example of how a morphological parameter can be used to differentiate between 3 different particle populations within the same sample. Intensity minimum, which is represented here on the x-axis, is a measurement of how translucent or opaque a particle appears to the MFI. The protein aggregate you see here on the right is highly translucent, and it scores very high for intensity minimum because it lets a lot of light through to the MFI. The silicon oil drop in the middle has a bright center but a very dark perimeter, so it scores lower than the protein aggregate for intensity minimum. And then finally, the air bubble on the left is highly opaque and scores the lowest for intensity minimum. So you can see here how this is the basis for how MFI is able to differentiate particle populations based on quantifiable morphological parameters. And this is just some images of some of the different particle types that can be found in protein formulations. Traditional particle analysis tools like light obscuration can't distinguish between different types of particles, which can give you insight into sources of contamination and stability. Using MFI enables you to identify what these particles are. So MFI provides a more sensitive detection method than compendial techniques like light obscuration and manual microscopy. So Since protein aggregates are often highly translucent, they can be missed by techniques such as LO and MM. And in this study that I show here of a protein sample, MFI was able to detect up to 10 times more particles than light obscuration and 100 times more particles than manual microscopy. The data here is a study done by the National Institute of Standards and Technology, showing the sensitivity of MFI based on its ability to better differentiate translucent particles from the surrounding matrix when compared to light obscuration. In this particular example, bead standards were measured in matrices of variable refractive index. The light obscuration measurements, which are represented by the blue series, are impacted much more strongly by the refractive index, which impairs light obscuration's ability to measure the bead size of 20 microns. The MFI measurements represented by the red series are far more robust to the changes in the refractive index and require only a small difference between the refractive index of the particle and of the matrix to achieve an accurate measurement. This was a study that was published showing the difference in sensitivity between MFI and light obscuration when looking at protein aggregates in the subvisible size range. So here you can see that MFI was significantly more sensitive than light obscuration in the number of protein aggregates that were detected. And here again is another study showing how MFI sensitivity is important for characterizing the stability of a protein. So in this case, they're looking at aggregation responses to stress, which can be important when developing a product, as this can translate into earlier indications of a protein stability. So here they compared the abilities of MFI and HIAC, which is a light obscuration technology, to detect protein aggregation in solutions of a monoclonal antibody that had been subjected to different forms of stress. In both cases, MFI was able to see an order of magnitude more protein aggregation than HIAC. So one of the key aspects of the MFI imaging technology is the use of depth of field and how that translates to data quality. If you look at the image here on the left, that image has a narrow depth of field. So that what's in focus is in a very narrow plane. The flowers that are in the foreground and in the background are not in focus and are very poorly defined. In the picture on the right, there's a broader depth of field, so that all the flowers are equally in focus and well defined. So how that applies to MFI is MFI uses an equal depth of field throughout the depth of the flow cell. What that means is that when compared with a narrow depth of field, this provides for much better imaging of all the particles in solution and better accuracy and precision in measuring all the particles in the sample. Another important aspect of the MFI is the sampling efficiency of its measurement. The MFI flow cell and optical path are designed to maximize the amount of sample which can be reliably imaged for particles. MFI is able to accurately measure 85% of the percent is not imaged due to non-laminar flow, where the fluid contacts the edges of the flow cell. That fluid flows at a different rate than the rest of the sample, so it can't be accurately characterized. But by providing an 85% sampling efficiency, MFI allows you to minimize the amount of sample that's needed to get an observation with high statistical significance. So here's this slide shows a comparison of MFI and another fluid imaging system, comparing the measurement of polystyrene spheres. And here you can see, particularly with the 5 and 10 micron beads, that MFI exhibits better precision and accuracy with sizing these beads. One important aspect of characterizing subvisible particles is having the right software tools to do the analysis of the particle images. Our analysis software, which is called MFI Image Analysis, includes powerful filtering techniques and also includes preloaded filters as well as user customizable filters to categorize and separate particles based on different morphological parameters. A classic example is differentiating things such as silicon oil from protein aggregates and other contaminants. So for cases where higher throughput is needed, we offer the Bot1 autosampler, which bolts onto the MFI and can be easily removed when you need to run the MFI manually. The Bot1 uses a 96-well plate format and has an automated pipetter that handles sample mixing, cleaning, and introduction of the sample into the MFI. It allows you to automate up to 90 samples per run, and it works with 2 mil deep well plates. Automation provides the advantage of being hands-free as well as having consistency in sample handling. So in summary, the MFI 5000 series instruments and the Bot1 autosampler provide a precise and accurate solution for detecting and characterizing subvisible particles in biotherapeutic products. Thank you to Marina for her presentation, and thank you all for your time, and I'll hand it back to Stephen.
Thank you, Dr. Edwards. Now we will begin the question and answer segment of the webinar, but once again, I would like to remind the audience that you can still send your questions in via the questions tab located directly below your webinar screen. So now our first question. What is your preferred method to measure concentration of subvisible particles, and what are the advantages and disadvantages of that method? Marina, would you like to tackle this?
So basically, I think I can speak about MFI that was presented here, but also can say that in some cases where the particles are very translucent and hard to basically get the contrast, or sometimes we're missing some of them, the good method to, as orthogonal, good orthogonal method would be culture counter. And we had some successes with those, and on the also prototype vaccine for tuberculosis, and I think that was also shared by webinar, and there is a publication on that. So I would just say that it's always good to double check the observations. However, both techniques have applications, and there are examples when the total numbers are very close. And if those are not, it means that you may have particles that not translucent, but they come from the process, and maybe more attention has to be paid to examine the differences. So I would recommend two orthogonal techniques for most part, but there are others like FlowCam, and you can even count some from other techniques, such as light obscuration. But as Bonnie mentioned, light obscuration is not really a sensitive technique, although popularly used because it's validated and it's listed in USP and European pharmacopoeias. So, thank you.
Thank you. Our next question is, do you have any examples of measuring particle sizes online or inline? If yes, what are the applications? Again, Marina, would you like to answer this?
So I would say that for some particles, I'm not really sure for proteinaceous because no experience just yet. But for the particles that contain adjuvants, one can also use focus beam reflectance measurement. And we came across this technique quite recently, but have some success stories with aluminum phosphate adjuvant and with absorbed proteins on aluminum phosphate adjuvant. And the results are consistent between what we see by laser diffraction and FBRM. So if the particles are, let's say, not very translucent, you may have success with those. However, we need more experience with that, but I would say FBRM definitely one of the options if one wants to move inline for the inline measurements using probes. I'm not sure, maybe Bonnie can elaborate on that, if there is any application for MFI, say for online, as a bypass to be attached for the vessels to be measured for the at manufacturing scale. Maybe Bonnie can elaborate.
Yeah, we don't currently have MFI a product for online, I guess, but that is definitely something that we've looked into, and we have seen some interest in using MFI online. But we don't currently have, like, I guess, an application in the field at this point.
Thank you. Our next question is it possible to use viscous solutions within the MFI? Bonnie, would you like to answer this?
Yeah, the MFI is, we rate it to be able to handle viscous solutions up to 20 centipoise. For solutions that are more viscous than that, we can't guarantee that it might not affect the flow rate through the flow cell and affect the data quality. So additional controls might need to be run in order to assure that the viscousness of the solution beyond 20 centipoise doesn't affect the data.
Thank you. Our next question, does the MFI have software that is 21 CFR Part 11 compliant?
Yes. So our MFI software consists of two parts that work together. So our MFI view system software is the software that runs the system, and then we have the MFI image analysis software that I mentioned. Both pieces of software are 21 CFR Part 11 compliant. Image analysis maintains that compliance if it's installed on the system computer. So you can export directly from MVSS, which is the view system software, to image analysis and maintain a compliant workflow.
Thank you. Moving on, next question is, what are approached for collection particles before running SDS page? Marina, would you like to answer this first?
Yes, so in our situation, we were quite lucky. We could just spin the sample at 4000 G and for 15 minutes and collect the pellet, basically remove the supernatant and collect the pellet. And then the pellet was resuspending with a buffer that we normally use for HSV2 vaccine candidate. And after basically add other constituents that you need for SDS page running, and then run the gel. Also, I have to mention the key thing was to add urea there, because you see pellets, so you have to resuspend, so it was also urea used. And after that, the gel was run. So we had two copies, as I mentioned. So one copy was used for Western blotting, and the other copy was used to excise bands. It was an old-fashioned technique. 2 years later, I can say that we can have same procedure without excising the bands. We can do it in solution. But at that point, yes, at that point, it was done in gel. So, but I also want to highlight that if you want to do microscopy, you have to collect it on filter. In some cases, you can do it on the paper filter. That works. In some cases, you really have to use metal filter. Metal filter varies. There can be even gold filter. The idea is just to extract the particles so it's solid enough for you to analyze after. I think that covers.
Thank you. Bonnie, do you have anything to add?
No, I don't think so.
No problem. Moving on, our next question is how concentrated of a sample can be run in the MFI? Bonnie, would you like to answer this?
Yeah, so it depends on the model MFI that's used. We have the MFI 5100, which sizes particles in the range of 200 to 300 microns, and then our MFI 5200, which sizes particles in the range of 1 to 70 microns. For the MFI 5200, we spec a limit of 900,000 particles per mil, and then for the 5100, we spec a limit of 175,000 particles per mil.
Thank you. Moving on, Marina, have you tried to measure subvisible particles in the presence of adjuvant?
Yes, basically that's another product. I have to say it's protein-based vaccine with adjuvant. It's called IC31, and we have a publication on that, and we have a webinar on that, so you have a choice. It's called HY31 vaccine against tuberculosis. That's a candidate we had. And first author is Sasmit-Muk. And me as well, so you can search by either one of us, and it's a Journal of Biomedical and Pharmaceutical Analysis, and that was published in 2018, June, I believe. So you can search it. It's free article, so you can download it. So yes, we had, and the techniques we used Mfi, by the way. I don't think it's part of publication, but we definitely use it to answer reviewers' questions. And we showed the occult counter that was successfully used for subvisible particles. We also did DLS, although DLS is really at the margin, only to confirm that we're in the right size range. We did also PALS, phase analysis light scattering. Why I'm mentioning that? Because for some subvisible particles, it's important to check the potential. And because this is your vaccine, really, that's overlap. So, yeah. Okay.
Thank you for your answers, and thank you, audience members, for your questions. Actually, we've got time for one more. In general, if you want to differentiate between air bubbles, silicon oil droplets, and protein aggregates, what morphological filters do you apply? As well as this, what sort of general values do you set? For example, aspect ratio below 0.85 to filter out most of the air bubbles and silicon air droplets. Would Bonnie like to answer this first?
Yeah, sure. There are a number of different morphological parameters that you could apply in this case. Some of this, there's a particular filter that we have pre-built into the software called the S-factor filter that was, I believe, developed it was published in a paper. And I think that uses a measurement of circularity and a couple other factors to differentiate between silicon oil and protein aggregates. So you can set different morphological parameters based on circularity, equivalent circular diameter, which is called ECD. Also the translucency, so the intensity minimum to measure the translucency of the particles, as I showed earlier in the presentation. As far as specific filters, there's more things more details that we could go into, and they're free to contact me directly if they want some assistance with setting up some of these filters.
Marina, do you have anything to add?
Not for this question, but for another question, I was thinking about whether we measured subvisible particles in presence of adjuvant. I think it's a little bit deeper than it was asked. So basically, if you have to absorb the protein after, so your protein or virus, to that matter, has to be absorbed, you have to measure at the pre-absorption stage, and then basically measure the particles or total count if you're interested for the adjuvanted. In our case, it was absorbed, and we measured the total particles in their morphology and the total count, concentration number, and so forth. So, and size distribution. So really, if the question is what happens to your product before absorption, you have to do it at the pre-absorption step. If that's really the angle of the question, I think that's worth to mention now.
Thank you for your reiteration. And once again, thank you, audience members, for your questions. Before we finish the webinar, I would like to ask our presenters, both Marina and Bonnie, if they have any closing remarks. So Marina, would you like to go first?
First of all, I would like to thank organizers once again. Thank you, Stephen. And this is a great opportunity to present with Bonnie. I really enjoyed it. And it's good to have more in-depth and more updates on Mfi because we're using it. And it's really important to know what other advantages the method brings, especially with the software where I can navigate between the type of the particles. And also say that I think more emphasis now is paid on morphology and sharpness of image. So anything that direction would be greatly appreciated. And I think, as was mentioned before, there is a need to bring it in line or online. That would be definitely a well-received trend, because there are questions to bring testing to the production and have it combined to streamline the whole processes. So I think you may find applications where people will receive it quite well. So thank you with that, and thank you, audience, very much for the questions. Very interesting, entertaining. I can see some of them related to LCMS, and I would like to refer back to the paper. And I think there is one more question related to aggregation modeling. Also, I would like to refer back to the manuscript reference that I provided, because that tells more in depth, and we don't have time to discuss it now. So, thank you.
And Bonnie, do you have any closing remarks for our audience?
Yeah, I just want to thank Marina for her work, and I'm glad to see MFI being used. In terms of using MFI inline, that's definitely something that we've had interest in looking into. So if there are audience members or people out there that are interested in doing that with MFI, I'd certainly love to hear about it and see if there's something that we could do to work forward on that. I just want to say thank you to everyone for attending, and thank you to all our customers out there. And thank you to Stephen and Biopharma Asia for hosting this webinar.
So thank you both. And I would like to take this opportunity to once again thank our presenters. That was Marina Kirkitadze, Head of Process Support and PAT Platforms, Analytical Sciences at Sanofi Pasteur, and Bonnie Edwards, Product Manager of Imaging and MFI at ProteinSimple. Thank you both for sharing your knowledge with us. I would also like to remind our audience that you can view this and other webinars on demand by visiting biopharma-asia.com/on-demand-webinars. And if you are watching this on demand, then please feel free to send your questions over to me at stephen.edwards@biopharma-asia.com, and we'll get those answers back to you. Thank you, everyone. Have a great day.